when you look at these targets, you'll see that there are some things that are very, very unique to prokaryotic cells, right? One being the cell wall. Now, I know we haven't talked a lot about bacteria structure, but if I'm putting it up here, Savannah, do you think that you have a cell wall in your cells? Good, you think correctly. And I think some of you that have probably had Bio 2, I know we did some basics with bacteria in there, so some of this you should be aware of. Those of you that haven't had Bio 2, right, trust me when I say your cells don't have cell walls. Does anyone remember some cell types besides bacteria that have cell walls that maybe you've encountered before? Plant cells, yes, good, plant cells. What else? Huh? Bacteria do, right? There's kind of one other big one that we'll actually maybe cover later this week. Maybe sometimes you've had them, what did you say, Jada? Fungi, right? Perfect, right? Fungi, right? Plant cells and bacteria. Those are some of the big ones, right? You don't have a cell wall. And so this is a good target, right? Because if you take medication that targets a cell wall, it should, for the most part, leave you alone. What that we talked about briefly on Friday, these all blend together when you're on your couch. On Friday, why is that medication still possibly going to have a negative effect on you, even if it's targeting cell walls and you don't actually have any?
Your normal microbiome.
Perfect, right? Your normal microbiome. So that word of dysbiosis, right? And so your body that have cell walls. And so that still could cause an issue, right? So it's still not perfect, right? Inhibition of protein synthesis. This is something that we typically talk about a little earlier, actually, except when I flipped it on its head, right? Can someone remind me from one of their basic biology classes what organelle, right, is important for synthesizing proteins? Ribosomes, yes. And it turns out that structurally, prokaryotic ribosomes are slightly different than our ribosomes. So what do you know? That actually gives them, or gives us, a very good target for medication. Okay. Does anyone know something that's kind of a little funky about our ribosomes and bacteria ribosomes? Why there might be some similarities actually? Who gave birth to you?
Mother.
Your mother. What else did your mother give to you that your father did not? Your mitochondria, right? And if you're not quite sure why that happened, just take it from me, right? This is how I explain the purpose of sperm and eggs, right? A sperm is a genetic rocket with fuel and a little bit of genetic material, right? It's a dumb missile, all right? The egg is where the magic happens. It has everything, all right? If you actually look at a sperm, all right, the mitochondria are right kind of all along the tail. And so it's only the head of the sperm that penetrates the egg and delivers genetic information, right? So you get your mitochondria, they're all your mom. And interestingly enough, the mitochondria has its own. And so mitochondrial identical bacterial ribosomes. Okay. Again, taking medication high doses will target bacteria, but eventually you'll start to see a toxic side effect because your mitochondria possess, right, the same type of ribosome. Okay?
You said ribosomal DNA is different than?
So the ribosomes, so again, this is a bit back to basic cell biology. If you talk about your ribosomes, most of your ribosomes are floating around in your cytosol or attached to your, what are they attached to? Yeah, they are, right? The rough ER, my favorite, right? And so the rough ER, all right, those ribosomes and the ribosomes in your cytosol are ribosomes that are in bacteria. How of a unique organelle, because contained within it are ribosomes that actually resemble bacterial ribosomes. All right, and we're not again here to have the evolution or macro versus microevolution kind of debate, but if we go the macro route, there's speculation that basically mitochondria at one point were bacteria that formed a symbiotic relationship with what we now know as eukaryotic cells. They decided that the marriage was good, and so they stayed together forever, right? Those of you that love plants, do I have any nature folks, environmental folks in here? You love plants, Alyssa? No, you don't. All right, but yes, that's all they're good for. No, just kidding. But if you like plants, right, one of the organelles that you've probably heard of is chloroplast. And chloroplast also, right, have their own set of DNA and their own set of ribosomes. And so we have the same kind of theory with mitochondria and chloroplast. They formed this relationship way, way, way back when, right? But again, it's still a pretty good target. Yes, ma'am.
The chat is saying that the mic's
Let me try to hold it more steady. But that's, I mean, I'll hold it right here. Savannah, as soon as you see it move, like kind of gasp or like scream, like. But if you do see me start to like, seriously, like throw something at me, okay? So we also inhibit nucleic acid synthesis, all right? Now, fundamentally, nucleic acid synthesis between a prokaryotic cell and eukaryotic cell are similar. What we will find out later in the semester, though, is that the speed at which a prokaryotic cell can do it is quite significant. We haven't mentioned it specifically, but some of you may recall that prokaryotic cells do not have a nucleus, all right? We do have a nucleus, so that basically means that nucleic acid, aka DNA and RNA, are freely floating around within the prokaryotic cell, all right? So that's kind of a unique target. Also remember, how many chromosomes do you have, Taylor? Do you remember? 23 pairs, right, or 46 individual. How many do you think bacteria have? Ah, she nailed it, right? Perfect, one. So they have one big giant. So there are unique features to nucleic acid, even though fundamentally, right, it's the same concept. They have the same base pairs as us. Replication, again, fundamentally is occurring similarly. Right? You have to replicate DNA. You have to turn DNA into RNA. You have to turn RNA into protein. So fundamentally, that stuff's the same. But as we go on throughout the semester, you'll see that there are unique differences. And these unique differences allow us to use nucleic acid as a target for these bacteria. All right, what do I have next? Metabolic pathways. Again, this probably, I'm trying to look at these, of the medications, this is the most limited. Why is this the most limited of the medications, right? There's not a lot of variations or varieties that are available here. Does anyone have a guess as to why that is? And I'm not trying to trick you, keep it simple. Sarah, why am I not making a bunch of medications that target the metabolic pathways of bacteria? Why do you think? The pathways are similar, right? A lot of them are identical or very, very close, right? And so this is not something that we have a lot of targets on. We'll talk about kind of, I guess there's two medications, but they're always kind of given together. So I just kind of consider it one medication that's given that goes after these metabolic pathways. So again, Sarah's right. There's a lot of similarities. And again, we mentioned like one of those first lectures. This is why bacteria or prokaryotic cells, right? Or not prokaryotic, bacteria or other microbial cells such as yeast, which are eukaryotic, are great research tools because metabolically they're very similar. That's great if you're trying to study the pathway. It's not great if you're trying to make a medication, but we still do have some stuff, right? And then interfering with cell membrane integrity. Now, truth be told here, right? Their cell membranes, in some cases, are really similar to ours, but again, there are a number of unique features to their cell membranes. If we talk about gram-negative bacteria, they actually have an extra cell membrane, which means they have an extra target for us. Okay. And so this is just I kind of like this diagram. This is on the PowerPoint that you guys have access to, and it just takes everything that we talked about, and it gives a little bit of a visual spin on it so you can see, right, what our medications are trying to target, all right? And this will be an important diagram for you guys to look at when we talk about antibiotic resistance here in just a little bit, because there's another diagram that looks similar to this one, but it's showing, hey, what do bacteria do, right, in order to resist, right, the effects of medications that we're taking. Okay, so I'm not going to spend time on this. This is what we just talked about, but it might help kind of, I don't know, maybe you put, oh yeah, nucleic acids, this is a cell wall, cell membrane, all right? Now, yes? Well, okay, so if we're talking about it being useful for like the bacteria, if we're talking about it being useful for the bacteria, that's an extra layer of protection for the bacteria. So it turns out that extra cell membrane that bacteria possess make it much harder for us to hit them with medications. All right, so it's protective, if nothing else, for the bacteria. All right. Now, you know, one of the things we haven't talked about, you know, much is if we talk about penicillin, right? And penicillin is probably, it's the antibiotic that we've all heard of. We mentioned it earlier in this lecture, or not in this lecture, but in lecturing on this chapter, right? So penicillin targets something called gram-positive bacteria, right? They lack that extra membrane, that outer membrane. So basically a gram-positive bacteria has a cell membrane just like you, and then it has a cell wall above it, all right? And penicillin targets that cell wall. It's the first thing that it touches. It's easy contact. Penicillin doesn't work on a gram-negative bacteria because the cell wall is sandwiched in between basically those two cell membranes. All right, so it's a very kind of unique protective feature that those bacteria can have. So if that doesn't help, Alyssa, let me know, okay? So the next diagram here, all right, is actually showing you the most commonly prescribed classes of antibiotics along with the most commonly prescribed antibiotics from those classes, right? And so when we talk about a class of antibiotic, basically what I'm showing you here, right? And we're going to talk about these in just a second. These different antibiotics here are going after some of those different targets, right? So like penicillins are going after the cell wall, right? You're going to have some antibiotics that are going after protein synthesis, right? And so these are kind of what we're targeting. And then these are the antibiotics from those groups that are commonly being prescribed, right? So you kind of have your main group of antibiotics that inhibit cell wall synthesis. And then within that group, you have a bunch of different kind of varieties. So that's kind of what this is showing. And charts like this is from the CDC, are just kind of important because they let us know basically what healthcare providers are using. One of the things that we'll talk about in antibiotic resistance, and I believe this is on the one video that I have on there for you guys to watch. So again, I'm not saying if you haven't done it yet, not a big deal, but please do it before the exam. But one of the things the guy on the video is talking about is how do we hold healthcare workers more accountable for antibiotic use? And so one of the things that some places are starting to do now, and not in an attempt to shame, but I think in an attempt to maybe make doctors realize or remind them how much antibiotics they're prescribing. Some hospitals are listing and then kind of posting, hey, you know, Dr. Taylor Moran, right, has prescribed this much antibiotics. And again, it's not to shame, but sometimes you don't realize, like, I don't know, if any of you have iPhones and you get like the weekly update that talks about your screen time. And sometimes I see mine and I'm like, oh my gosh, I had no idea, right? It felt like it went really quick. And I think that kind of happens sometimes with healthcare providers and antibiotics, right? They're doing their job, right? And they're doing it well. And they're like, antibiotic, right? They're trying to help their patients. And they don't realize until it's kind of laid out there for them that, oh, hey, you know what? Like I could have probably cut back here or maybe even tried this antibiotic because it wouldn't have required as long, and so that's what a lot of this information is for, is not to shame, but to kind of bring to light kind of what's going on. All right. So let's start to talk about some of these, right? So amoxicillin, right, which again is an antibiotic that you guys have probably had at some point in your life. If you haven't, you probably will have it at some point in your life, as long as you're not allergic. Allergic to, like presents as hives. All right, and so they see the hives, and then the doctor knows, okay, switch to a different antibiotic. All right, so typically, not a antibiotic allergies are not a huge deal as long as they're taken seriously early on. All right, so again, it belongs to the penicillin family. As I mentioned, the penicillin family is trying to inhibit cell wall synthesis. All right. Now, again, when we talk about inhibiting cell wall synthesis, typically that's going to be more effective on gram-positive bacteria, right? Because they lack that extra outer membrane. But, right, there are some cell wall inhibiting antibiotics that still can target gram-negative. So please don't think that that cell wall inhibiting antibiotics only work on gram-positive. That's typically where they're most effective, but we do have some cell wall inhibiting antibiotics that can still work on gram-negative bacteria. All right. Now, amoxicillin is typically given with, I know it's kind of like an awkward situation for me, this clavulanic acid, right? Clavulanic acid is a beta-lactamase inhibitor, right? So I didn't mention this, but cell wall inhibiting antibiotics, one of the major groups in there are basically those antibiotics possess something called a beta-lactam ring. So it's a structural feature that almost all cell wall inhibiting antibiotics share. So what do you think beta-lactamase is? Just based on the name. Yes, it degrades beta-lactam. Remember, one of my favorite rules of biology, usually an ASE is an enzyme, and usually that enzyme's breaking up whatever's in front of it, right, in the name. So bacteria, and we'll talk about this in antibiotic resistance, can produce enzymes, right, that break down the antibiotic that you're taking. In this case, a beta-lactamase, right, is produced by bacteria. It breaks down the amoxicillin maybe that you're giving them. And so what we have some medication, right, is with some medications we give an inhibitor to beta-lactamase along with the antibiotic because it makes the antibiotic more effective. Now we'll talk about here in just a little bit, we'll see that some antibiotics, just based on their structure, are more naturally resistant to beta-lactamases, all right? So then you don't have to give the inhibitor, right? It just kind of depends on the antibiotic. But this basically allows you to get more bang for your buck, all right? It makes that antibiotic more effective, and then it also, right, lowers the, might have to give out that beta-lactamase inhibitor. Okay, yes, ma'am. A lot of beta-lactamase, well, that bacteria then may be like a gram-negative, and one of the reasons it doesn't have beta-lactamase is because it doesn't need it because the antibiotic can't get to it, right? And so, and I'm not saying you were saying this, but I don't want you to think, well, then just give somebody, a bunch of this antibiotic because the bacteria can't fight it. Well, more than likely the bacteria probably can fight it. There's just different mechanisms. And so kind of file that one away though, because that's a great point and a great question. You'll see when we get to antibiotic resistance that, again, there are a lot of different features that are going to allow them to resist a lot of different things that we throw at them. Okay. So again, you've probably had some of these issues, hopefully not all of them, but if you've had an ear infection, right, this is pretty common. It can be used for urinary tract infections, all right? You know, which those of you that take an ANP2, I know I at least mentioned this to my group, right? This is much more common in females, right? UTIs, and that's because the urethra, right, is much shorter in females, all right, than it is in males. And so males have bacteria in the urethra, right, but it has a lot farther to travel, right, than it does in females. And so there's a greater chance that infection, right, can move up the urethra of a female's and then into the kidneys. So it's just more of an issue in females. Same basic concept for ear infections, right? When you're a kid, you have your eustachian tubes here that connect to your pharynx or your throat, right? When you're small, they're small, which means bacteria can travel up them much more easily. So when you're a kid, you're more prone to ear infections. As you grow, your eustachian tubes grow, right? Bacteria can't travel as far up, or it takes them much longer, and you're able to fight them off and not have as many ear infections when you get older. Yes, ma'am.
And antibiotics, something that happens in an individual, or is it just something that happens bacteria in general when you get infected?
Both.
Okay.
Right. So we'll do a little sneak peek. So antibiotic resistance, right? The issue becomes, and you know, and again, I guess you could make the argument kind of chicken or egg, but either way it works. And so what would happen is I'll pick on Michael because I can. I know he can take it, right? If not, right, he'll come after me. But so like let's say Michael, right, goes in and he's not feeling well, and they've determined it's bacterial, and they say, hey, Michael, we'll take 2 pills a day for 10 days. Okay, and Michael's like, yes, I'll do that. All right. And then Michael starts feeling better on day 4 or 5. And so then he's like, I don't need to do this anymore. All right. And so the issue here, we'll just skip way ahead. We'll come back to all of this, I promise. So then the issue is that, you know, truth be told, in most of you, the bacterial population as far as susceptible and resistant is going to be somewhat mixed. But in the most cases, the susceptible, right, bacteria is going to be the majority of the bacteria. There's not going to be a lot of resistant bacteria. And so what happens, the reason that Michael starts to feel better immediately is because those first couple of days, those susceptible bacteria, they fall like flies. All right, they just go down. There's no resistance there. Now, you know, resistance, the way I kind of like to look at it is if you've ever watched, I don't know, an action movie, which I know is maybe not the most realistic representation, but, you know, you see them when they have their bulletproof glass, right? And truth be told, if you shot at that bulletproof glass long enough in the same point, right, you would eventually punch through it, right? And so bulletproof glass is resistant to bullets to a certain extent. So we kill off those susceptible bacteria, and Michael, the only ones that are left are the resistant ones, all right? So that extra 5 to 6 days of antibiotics is for those bulletproof, right, those resistant bacteria. And so you keep on pounding them and pounding them and pounding them until you kill them. The whole while, Michael feels better because the number of resistant bacteria compared to the susceptible bacteria was really small. And so, you know, you're basically saving that last 5 to 6 days of medication to go after a few bacteria that may be left, all right? Now, what happens is Michael, right, though, he stopped. So he killed off all of the susceptible bacteria, and he now has, right, a few resistant bacteria left. What we haven't really talked about yet, but some of you probably know, is that bacteria can grow at exponential rates. And so on day 4, when Michael stopped his medication, and let's say he had, you know, only 10 bacteria left that were resistant, well, by day 10, when he should have been finishing his medication, right, that could be in the thousands, it could be in the millions, all right? Now, when Michael goes back to get treatment, the antibiotic that he used doesn't work, or he has to take even more. And so then the problem is, Jennifer, a couple of things is that, let's say Michael is hanging out, I'll I'll throw myself under the bus, right? Michael comes to hang out with me because we're watching a special on Netflix about beard growing competitions because he's felt inspired by me, right? And so, we hang out, and then he passes some of that bacteria on to me. So it's a bacteria that developed in him that then comes to me. Now, to add even more kind of complication to this, what we see here is that when bacteria get stressed out and when bacteria start to die, they have this incredible ability to share information. And so what gets really scary is that the resistant bacteria that were in Michael, right, what we're starting to realize is that maybe that bacteria does eventually get killed by the medication, but when it dies, its DNA gets released. And then there's another bacteria in the area that picks up that DNA that contains that information, right? So all this information is contained within genes, right? There are genes, right, aka that become proteins that allow these bacteria to be resistant, all right? So then that information starts getting shared among other bacteria, right? And so you kind of have this, it just becomes this big cycle, all right? And then unfortunately, right, again, when Michael has all those resistant bacteria, what's our first go-to, our first solution? More antibiotics, right? So then you kind of start this vicious cycle that keeps on going and going and going. All right. Which one am I going to go this way? So here we'll talk about this really quick, and then we'll kind of pick up the pieces about specific medications. But these are, right, the basic ways that bacteria, right, are becoming resistant, all right, or are resistant to the medications that you take, all right? So one thing is some bacteria, right, and this would be gram-negative bacteria, have something called porons, right? And when you hear a poron, think a pore. It's a big opening, right? And that opening is important because that opening allows things to cross that extra outer membrane that they have, all right? Typically, when we think of porons or we think of a pore, we think of something that's just a big hole and things go in and out. And that's actually not true. Bacteria have a unique ability to regulate those porons, to close them or open them. And so what we see is that gram-negative bacteria, right, in the presence of medications will close their porons, right? They'll close that route in that allows things to pass that outer membrane, okay? Related to that, we also see things called efflux pumps. And basically, some bacteria contain genes that allow them to assemble a protein pump that pumps out medication. So it'd be like if I put Hannah, right, in a boat, and then I pulled, right, if you're in a boat, you'll see there's plugs, especially like in the small aluminum bolts. I pull this plug that's in the back, and then water starts to come in. What I also, though, have given Hannah is something called a bail bucket. And so as that water comes in, Hannah just bails it right out. And that's what an efflux pump does. So that medication goes in, the bacteria sees it, and they pump it right back out. Okay. We also see, right, that bacteria are able to alter the shape or the structure of whatever that medication is targeting. All right. So they basically disguise it. All right. Again, I'll pick on Taylor one more time. So if I say, okay, medication, kill everything in this room that has a red shirt on, all right? So then Taylor, right, being of above average, we'll say well above average intelligence, just says fine. She pulls a black sweater over her red shirt, right? And so she has changed herself, right? Now my medication no longer recognizes her. And that's what some bacteria can do. A simple alteration of the structure makes it unrecognizable for the medication, right? Then we've already talked about something like beta-lactamase. So we can produce an enzyme that goes after the medication and it destroys it or neutralizes it before it can reach its target. Yes, ma'am. It can do both, right? So that's what I'm saying. So the bacteria can alter the structure of the antibiotic target, right? So what you're targeting, right? So what I'm targeting, so if Taylor's the bacteria, right, she changes her outfit, right? She alters herself so that I can't recognize her, all right? So that's one way. The other way, though, the other thing, again, we'll just use Taylor, that Taylor could do, is she could pull out her antibiotic gun, right, whatever that is, and she could shoot the antibiotic before it got there, right? So you can change the target, or you can neutralize the antibiotic before it gets there. All right. Am I forgetting one? What other one did I miss? I feel like I missed one. I can't see it. I didn't angle that towards me. Isn't there 4? I did 4. Isn't there 5? What one am I missing? Oh, there's only 4. Okay, good. Man. It's just like a little unnerving when you can't quite see everything that you're talking about, right? And so this is what bacteria are doing, all right? And if you don't finish your medications, right, the bacteria that are more resistant then become the dominant population. If you don't finish your medications, right, and maybe you've killed some of the resistant ones, but they've shed some of their genetic information, other bacteria can pick that up, incorporate it into their genome, and now you took something that may have been susceptible, but you've allowed it to be resistant, right? And so this is what we're starting to see. This is a really big concern in the medical field, right? And again, we can talk about, I'll see if I can pull it up, right? It was a couple of years ago, but I think I already mentioned to you, some lady goes to India, breaks her leg, gets surgery there. By the way, if you can avoid surgery in India, based on reading this article, I think you should. She gets a bad bacterial infection. She comes back to the United States. They try every antibiotic that we have available at the time in the United States on her, and none of them worked, right? And she ends up passing away, right? And so this is becoming a big deal. That's not the norm, but those cases are starting to kind of tick up slowly. All right? All right. It's not all doom and gloom, right? Most of you, I hope all of you, are much smarter than me, and maybe a few of you will devote some of that brain power to this, or maybe some of you will devote your brain power to this via the avenue of public health, right, and educating people. I don't care how you do it, just do it, because I like my antibiotics when I need them. All right, so get out of here. Have a great day. As your exam scores get posted, which again I'm rolling through, have about, check them off if you're concerned, right? Email me, set up a time. I'll probably put a note on my door. I probably won't be here in the office for office hours, but I'll be willing to meet via Zoom during my office hours. This is just a little uncomfortable for me. So I'll probably go back to my couch when I'm not teaching, but I'm totally still willing to meet during those times. Okay, so please don't be afraid to reach out via that avenue. Buckner. Oh, yeah. I'll look into that and I'll send out an email. Those of you who were supposed to present last week, you two, can you guys be ready to go Wednesday? I'll need to check this week. You're presenting Wednesday? Okay. We might just all go Wednesday and just kind of get it out of the way it has to normalize at some point. So if you guys are okay with that, you're okay for Wednesday, you two are okay for Wednesday, then we're going to go for that on Wednesday. And I'll look at those articles and let you know when those need to be done. Holmes, what do you want?
I was supposed to do it today, so I can do it Wednesday.
You're supposed to do it today?